Hi, and welcome back to Chapter 5. Let's take a look at how things move in and out of cells with a little help from some proteins. So remember before we were talking about passive transport, and we talked about just how things moved across a membrane. There is some form of passive transport that is aided by proteins. And if it's passive transport, again, that's just diffusion and osmosis moving along its concentration gradient. But if it's helped by proteins, we say that's facilitated diffusion. To facilitate just means to help. So with facilitated diffusion, transport proteins kind of speed up the movement of molecules across that membrane. We talked about channel proteins before being like little corridors that allow very specific molecules or ions to cross the membrane. We even talked about aquaporins before. That's the kind of channel protein that helps water move across the membrane. We briefly mentioned ion channels, and those ion channels can actually open or close. Like they have kind of a gate on them that opens and closes and lets certain ions in or out. Those ion channels are also known as gated channels. Carrier proteins are the kind that go through those changes. I gave you the example before of the ketchup packet, right? It's going to change plate shape and allow things to go across. We say it translocates things across the membrane. The shape changing actually can be triggered by binding and release of the transported molecule. So that's interesting. The molecule itself causes the packet, right, the carrier protein, to change shape. But even though we've got a change of shape here, there's no energy input required for this to happen. So just kind of looking here at two types of transport proteins that can carry out facilitated diffusion. We've got our channel protein that's it's almost like a straw here that just allows things to go through one way or the other. It is real specific though. It only allows certain things to go through. And then we have our channel protein. And remember, channel proteins change shape. The solute binds to it, and that binding is what causes the channel protein to change its shape and allows the thing to move along. So far, everything that we've talked about is passive transport. Even facilitated diffusion is a form of passive transport because there's no energy required. But active transport is different. Active transport uses energy to move that solute against their concentration gradient. In other words, we're not doing diffusion anymore. We're not going down our concentration gradient. We're going against it. We're going from a place where there's not much solute to where there's already a lot of solute. I don't know. It's kind of like maybe putting the ketchup back in the ketchup packet. It takes energy. So that's active transport. Moving against the concentration gradient. This allows cells to maintain a concentration gradient different from their surroundings. This is so important. Living things don't match unit for unit whatever's outside. It's got to maintain this special environment. And so it requires energy, and most often that energy is in the form of ATP. You've probably heard of ATP before, and we will be talking about it extensively in upcoming chapters. But for now, we can say that ATP provides energy. One example of an active transport process is the sodium-potassium pump. Anything with the word pump in it, it's going to be moving against a concentration gradient. In the sodium-potassium pump, you're exchanging sodium, which is Na+, for potassium, which is K+, across the membrane. Let's take a look at what that looks like. So the sodium and potassium pump works like this. Notice that this is a carrier protein. The sodium binds to the pump, and that causes the pump to change its shape and release the sodium. At that time, the potassium binds to the pump, but it can only do this. Notice it can only do this if ATP is used up. So phosphorus is attached to that, releases the sodium. Then that phosphorus releases, and that allows the potassium to bind to the protein pump, and that allows the change of shape to happen, which allows the potassium to be released against its concentration gradient. That's the sodium-potassium pump. It's only active transport because, number one, energy was used, and number two, things are moving against their concentration gradient. If you want to find out more about how this works, you should check out the resources available from your book. There's one on active transport, and there's another one on the sodium-potassium pump. So a quick compare and contrast between passive and active transport. You can have diffusion, and remember osmosis is just a special kind of diffusion, directly across the membrane. Diffusion usually happens with hydrophobic molecules, and it happens at a pretty slow rate. It's hydrophobic, but they're also pretty small molecules. If it's larger or if it's hydrophilic, it's going to need a little help, and we call that facilitated diffusion. With facilitated diffusion, you can either have a channel protein like you see here, or you can have a carrier protein like you see here. With active transport, we are moving against our concentration gradient. There's a lot of these diamond-shaped molecules here, and we're trying to pump them against their concentration gradient into an area. Oh, I'm sorry, I've got this backwards. My arrows are backwards. The diamond shape are moving in this direction towards an area where they're already highly concentrated. And the yellow spheres, those are moving against their concentration gradient. They're being pumped into an area where there's already a lot of them. Ion pumps help maintain membrane potential. And membrane potential is actually, it's a term of electricity. Voltage. We're talking about electricity that is maintained by having different ions in different locations in the cell or outside of the cell. So it's maintained by having anions or cations moving those across the membrane. If you don't remember what anions and cations are, you might want to review your notes from chapter 2. But specifically here, as we're focusing on how ion pumps maintain this membrane, if you have two combined forces, collectively we're going to call this the electrochemical gradient. These two combined forces are causing this, driving this diffusion of ions across the membrane. There's the chemical force. This is the concentration of those ions, and there's the electrical force. This is about the membrane potential. It's about the charge of those various ions and how that moves across the membrane. So what we're going to be looking at in the next picture on the next slide is an electrogenic pump. An electrogenic pump is a transport protein that generates a voltage across a membrane. The sodium-potassium pump that we just looked at is a hugely important electrogenic pump in animal cells. If we look at plants and fungi and bacteria, the main electrogenic pump is something called a proton pump. We are going to be looking at proton pumps a lot later, so let's take a look at, just really briefly, one right now, and then we'll be able to focus on this when we get to it eventually in the chapters where we talk about mitochondria and we talk about photosynthesis. So this is a proton pump. A proton pump moves protons. And remember, protons have a positive charge. And the way that we depict protons in most biology books is by thinking about hydrogen ions. Hydrogen ions they only have one proton, and since an ion is lost its electron. So this hydrogen ion, this proton, is going to be moved across across the plasma membrane against its concentration gradient from an area where there's not very many of them to where they're already highly concentrated. Now, that's that idea of that chemical force, right? The chemical force is just the presence of the proton, but the electrical force is the fact that this ion that has a positive charge is being moved to where there's already a lot of positive charges. So on this side of the membrane, we have a lot of positive charges, and we have a higher negative charge on this side of the membrane. Another thing that we can look at when we're looking at active transport is something called cotransport. Cotransport happens when a transport protein basically has two things that work together. So we say they're coupled. And the transport protein couples diffusion with the opposite of diffusion, right? Active transport. So it's using the energy of something flowing downhill. I like how this is downhill is in quotes. I'm going to give you an example of this more physically in a second. But diffusion kind of happens downhill. Things go down a concentration gradient. It uses that energy to force something to move uphill, a second solute to move uphill against its electrochemical gradient. A good way to think about this is thinking about how water flows over a dam. And like that water flowing over a dam creates a waterfall. And if you put like a water wheel in the waterfall, you could use that wheel to move something else, like maybe I don't know if you're familiar with this, but a grindstone to grind up wheat or corn or something like that, like a mill. So coupling the downhill diffusion of one solute with the uphill transport of another, that's cotransport. If this isn't making sense to you, let's take a look at this picture. Here we see cotransport, which is active transport driven by a concentration gradient. Remember how in our proton pump, we pump things into an area of higher concentration? Well, we can then allow those protons to diffuse back into the inside of the cell, going down the concentration gradient, going from an area where they're highly concentrated to where there's less concentration. And that's natural, right? That's normal. Things move down. But that gives like a little energy, like water over a waterfall. We can use that to bring in other solutes that we want to bring in. In this case, we're bringing in sucrose. Bringing in this lovely sucrose so we can have a high concentration of sucrose inside the cell. Let's cotransport using this diffusion to allow this to be actively transported into the cell. We, of course, did use up energy earlier when we were using that proton pump. The last big concept that we're going to talk about in this section of the chapter is bulk transport. Bulk transport is when large molecules move in or out of the cell through a process of exocytosis or endocytosis. And I'll define those for you in a second. But remember that water and small solutes can enter or leave through the lipid bilayer by means of transport proteins like aquaporins. Large molecules and like polysaccharides or large proteins have to be kind of engulfed in vesicles. And it's that engulfing of vesicles that we're going to be talking about. Engulfing is going to require energy. Let's talk about exocytosis first. Exo means exit. So in exocytosis, transport vesicles, maybe ones that have been butted off from the Golgi apparatus, they're going to migrate or move towards the membrane, fuse with it, and release their contents. Lots of secretory cells use exocytosis to get rid of their products. And a secretory cell or a secretory cell, you can pronounce it either way, secretes things like hormones and or other things along those lines. They're putting the products outside of the cell, secretory cells. With endocytosis, endo like into, and cyto meaning cell, with endocytosis, things are coming into the cell. So the cell will literally make new vesicles by engulfing the object and forming these little vesicles that have originated from the plasma membrane. I'll show you a picture of this in just a second. But for endocytosis, it's basically the opposite of exocytosis, and different proteins are involved. There are 3 types of endocytosis. There's phagocytosis. Phago means to eat, so literally cell eating. This is where you bring in food particles like sucrose. There's pinocytosis, and pino means water, so this is cell drinking. And then there's a really cool one called receptor-mediated endocytosis, which I'll show you a picture of in a minute. Let's take a look at endocytosis. So each of those 3 are demonstrated right here. First is phagocytosis. And with phagocytosis, you basically are engulfing this object with the plasma membrane and bringing it inside the cell, and notice that you have this object is still surrounded by plasma membrane. Again, we could think about this as a food particle. Remember, phago means to eat. Pinocytosis is just bringing in lots of water. It might also grab some other solutes, but the vast majority of what it's bringing in is water. And this is a coated vesicle. It's got a coat of proteins around it, and that is the region of a cell that will do a lot of cell drinking or pinocytosis. The last one is receptor-mediated endocytosis. Remember we talked about receptors when we were talking about the different proteins that can be found on a plasma membrane. And so here we have a bunch of receptors, and they bind on to these solutes, and then we get a vesicle forming here, but specifically on the inside of this vesicle, all of those solutes are attached to receptors. So receptor-mediated endocytosis. I'll show you another version of this in a second. Specifically, we're going to be looking at how cholesterol is used to be brought into the cells through receptor-mediated endocytosis. Cholesterol, it's very important to be pulled out of the bloodstream, and cholesterol does travel in these particles called low density lipoproteins, or LDLs. LDLs are your body makes them, and it's actually a form of, it's inherited, and it can cause something called hypercholesterolemia, which means you got too much cholesterol. It's just a fancy word to say your cholesterol levels are high. And this happens when people don't have enough LDL receptor proteins on their cells. So, or maybe they're missing completely. So if your body's not making those LDL receptor proteins, then when you eat something high cholesterol, your cells can't pull that cholesterol out of the bloodstream. It stays in the bloodstream, accumulates in the blood, and that contributes to arteriosclerosis, which can lead, of course, to heart attacks. So in normal cell, you've got all of these LDL receptors, and they bind to the LDL and bring them in through receptor-mediated endocytosis. If you've got a mild form of the disease, you're not making as many LDL receptors, but you still have some, and so you're pulling out some of those LDLs. And if you have a severe form of the disease, you're not making any of those LDL receptors, and you will not be able to take the LDL out of the bloodstream, and that would be very, very unhealthy. So that's why receptor-mediated endocytosis is so very important. Well, we're now done with chapter 5, part 3, and we're going to move on to part 4, where we're going to learn about cell signaling.